This is the first 60,000 letters of chromosome, one of the draft public domain human genome. It looks like gibberish, but these characters represent the actual physical structure of the molecule of inheritance. A sequence of these letters would be equivalent to a random sequence of Greek or Sanskrit words, but the language of life is universal. Every single human being alive today has this exact same sequence with only minor variation. It's here in this string of letters that we find the essence of what makes us us. And if we learn how to read it, we might just learn more about ourselves than we ever thought possible. Each character here is a letter of a four-letter alphabet: A, C, G, and T. These are not the only possible letters, but they are the ones used by nature. In fact, these letters are chemically identical to those used in the 1950s to encode messages during the Cold War. The four letters of DNA may have been chosen arbitrarily by scientists when creating the genetic code, but they are far from random. With these four letters alone, every possible combination of codons can be represented. Codons are groups of 3 letters. Since there are 4 letters in the alphabet, there are 4 or 64 unique combinations. More than enough to encode the 20 amino acids needed to make proteins, which are the building blocks of everything living. It's also worth noting that every possible combination of 5 letters is found in this sequence. This is not quite the way the information is stored in the real world, however. In the cell, this sequence exists as a double helix. 2 strings of these letters, each paired up with their complementary partner. A always pairs with T, and C always pairs with G. Like 2 old friends meeting after a long time, they recognize each other immediately. They do this by forming hydrogen bonds with each other. And it's these bonds that hold the 2 strands together. If you want to split the helix into 2 separate strands, then you have to break these bonds. And that requires heat. The double helix is stable and strong, but not indestructible. The reason we use these letters instead of something else to represent the bases is because of some ancient candy. You see, it was the color of these candies that inspired the symbols used in molecular biology. But why these particular letters? Why not say Bob, Sue, Tom, and Alice? The answer lies in a bit of history. The man who cracked the genetic code for humanity didn't have the internet to distract him. Instead, he spent his time playing with candy. And I don't mean that figuratively. His name was Robert Holley, and he really did spend a lot of time playing with candy. He was trying to figure out the genetic code, but he wasn't just sitting around waiting for it to magically appear. No, he went out and hunted down the molecule responsible for transferring genetic information from the nucleus to the ribosomes where proteins are made. That molecule is called transfer RNA, or tRNA. In order to study tRNA, he had to isolate it from animal pancreas tissue. And while the methods used back then were incredibly primitive compared to today's technology, the results were revolutionary. When he analyzed his sample, he discovered a strange thing. The molecule was shaped like a clover leaf, and it contained a specific sequence of these mysterious 4 letters. These sequences were actually the answers he was looking for. In fact, what he found was the key to understanding how genetic information could be transferred from the nucleus to the ribosome. And so the 4 letters became the universal language of genetics. They were given names based on the chemicals they represented: adenine, guanine, cytosine, and thymine. And with these 4 letters, the language of life can be written. Here is the first codon, AUG, that's methionine. Next, U, that's phenylalanine. Then UCU, that's serine, and so on. Each group of 3 letters represents 1 amino acid. These amino acids are the building blocks of proteins, which are molecules that do most of the work in the body. Proteins form long chains, and it's the sequence of these amino acids that determines how the protein will fold and what shape it will ultimately take. This is important because the shape of a protein determines its function. But why does it matter that this sequence was discovered by accident? Well, sometimes things happen for a reason. This sequence was the first to be deciphered, but it wasn't the last. Using this method, the entire genetic code was cracked in just a few years. And soon after, we were able to synthesize new DNA molecules with any sequence we wanted. It's no exaggeration to say that this discovery revolutionized our understanding of life itself. It showed us that all living things are made of the same stuff, just put together in different ways. And that's why I want to share this sequence with you. It's not just a pretty picture or a cool bit of trivia. It's a symbol of our ability to understand the natural world around us. It's a reminder that even in the most complex systems, there's always a simple, underlying order. All we have to do is look for it. Now let's go back to the double helix. Remember how I said that these letters exist in pairs? Well, here they are lined up side by side. At first it looks like random noise, but if you squint your eyes, you can see a pattern emerge. There are two intertwining ribbons of letters, and each letter on one ribbon is paired with the letter on the other ribbon. The two strands run in opposite directions, but they're held together by hydrogen bonds between the base pairs. Now I want you to do something. Close your eyes and imagine that you're a scientist in the early 1950s. You've just discovered a strange new molecule inside the nucleus of a cell, and it looks like this. You know that this molecule carries genetic information, but you don't know how. So you start to investigate. You notice that the molecule seems to be made up of smaller parts, and you isolate them. You find that there are 4 different kinds of these parts, and you call them A, B, C, and D. You also notice that these parts seem to fit together in a specific way. A always pairs with T, and C always pairs with G. You don't know why, but somehow these pairs seem to be held together by a special force. If you were this scientist, what would you do next? Well, probably the first thing you'd want to do is figure out how these letters combine to make words. And the basic unit of language is the codon. The scientists who were doing this research in the 1950s knew this, but they didn't know what the words were. They just knew that each word was 3 letters long. So they started trying out different combinations until they found one that worked. They were looking for a word that coded for one of the 20 amino acids used by cells to build proteins. Eventually, they found it. The first word they cracked was AUG. They didn't know what amino acid it coded for, but they did know that it was a start signal. Without this signal, the ribosome wouldn't know where to start building the protein. Once they knew the first word, they could move on to the second word. And once they knew the second word, they could move on to the third word, and so on. Before long, they had cracked the entire genetic code. They had figured out which words coded for which amino acids. This was a monumental achievement. It unlocked the secrets of how genetic information is stored and transmitted. It also gave us the ability to manipulate DNA and create new genes with new functions. But it wasn't easy. It took years of hard work and countless experiments to crack the genetic code. But it was worth it because now we can understand the language of life. We can read the sequence of amino acids in a protein and predict how it will fold and what it will do. We can also write new sequences and create new proteins with new functions. This is the power of the genetic code. And it all started with 4 letters. Why are these letters important? Because they're the basis of life itself. Everything living on Earth uses these 4 letters to store its genetic information. We share these letters with bacteria, fungi, plants, and animals. We are all made of the same stuff. It's just the way these letters are put together that makes us different. So the next time you see a sequence of letters, don't just ignore it. Take a closer look. See if you can find the patterns and beauty hidden within. Because these letters are more than just random characters. They're the language of life. And once you understand that language, you can understand everything.